Hello everyone. Now we will discuss about the mechanism of cell injury. On the basis of nature, duration and severity of an injurious stimuli, there will be a cellular response, and depending on the nature, duration and severity, this response could be reversible or irreversible. Consequences of cellular injury depends on the type, state and adaptability of the injured cells. After exposure to injurious stimuli, there are multiple interconnected mechanisms got activated and result into the cell damage. Now we will discuss them one by one. First is mitochondrial damage. Whenever there is hypoxia or decreased blood supply along with the decreased survival signal, this results into the mitochondrial damage. After mitochondrial damage, this results in decreased oxidative phosphorylation or decreased ATP formation. As we know, production of ATP mainly takes place in mitochondria. So there is decreased ATP production, and this ultimately hamper all the energy dependent function and result into the cell damage. Another mechanism which takes place in mitochondrial damage is increased production of reactive oxygen species, and they damage protein, lipids and nucleic acids and result into cell damage. And this form of cell damage is necrosis. The third mechanism of cell injury in mitochondrial damage is leakage of mitochondrial proteins. They are pro-apoptotic in nature and stimulate the apoptosis, which is also a form of cell death or cell injury. Now we will see another mechanism of cell injury is membrane damage. Reactive oxygen species or free radicals, they directly damage the lipid of cell membrane by lipid peroxidation. Hypoxia result into decreased phospholipid reacylation or synthesis, and increased cytosolic calcium activate phospholipase, which ultimately result into the increased phospholipid breakdown. By all these three mechanisms, there is loss of phospholipid and result into the cell membrane damage. Increased cytosolic calcium also activate the proteases, which damage the cytoskeletal proteins and directly damage the cell membrane. Increased phospholipid breakdown also result into the lipid breakdown products. They are also harmful for the cell membrane integrity. Once cell membrane damage takes place, this can be result into the mitochondrial membrane damage, plasma membrane damage or lysosomal membrane damage. In mitochondrial membrane damage, there is creation of the transition pore, and as we know, whenever there is damage to mitochondria, decrease in ATP production or release of the pro-apoptotic proteins result into the apoptosis. In plasma membrane damage, loss of the selective permeability of membrane, whenever this membrane damaged by any reason, result into the swelling of the cell and ultimately cell death. In lysosomal membrane damage, there are release of the acid hydrolases which damage the cell by necrosis. So this is the mechanism of membrane damage, how membrane damage takes place, what are the causative agents like reactive oxygen species, hypoxia, and increased cytosolic calcium. They ultimately damage the cell membrane or membrane of the organelles like mitochondria and lysosome, and ultimately result into the cell injury by apoptosis or necrosis. Another mechanism of cell injury is damage to DNA. Reactive oxygen species, anti-cancer drugs, radiation or chemical agent, they directly cause damage to the DNA. Whenever there is DNA damage, this activates the P53 gene. P53 is a tumor suppressor gene. P53 activate cell arrest in G1 phase, and it also stimulates the DNA repair mechanism. If these repair mechanism fails, then P53 stimulates the apoptosis by mitochondrial pathways. So this is how P53 protect a cell from transforming into a malignant one. Another mechanism of cell injury is disturbances in calcium homeostasis. Normally, intracellular calcium stored in endoplasmic reticulum and mitochondria. Whenever there is an injurious stimuli, this damage these organelles, endoplasmic reticulum and mitochondria, and increases the level of intracellular calcium. Intracellular calcium whenever increase, this result into the increased mitochondrial permeability and opening of the transition pore and decrease ATP production. On the other hand, intracellular calcium activates the various enzymes, namely ATPase, phospholipases, proteases, and endonucleases. ATPase again cleaves the ATP and further decrease the ATP production or decrease the ATP. Phospholipase degrade various phospholipids. Proteases, they cause disruption of membrane and cytoskeletal proteins, and endonucleases responsible for the nuclear damage. This phospholipases and proteases collectively result into the membrane damage. Now we will see another mechanism of cell injury, that is endoplasmic reticulum stress, or also known as unfolded protein response. Whenever there is metabolic alteration, mutation in proteins and chaperones, viral infection or chemical insults, this will increase the misfolded protein. Normally endoplasmic reticulum fold these misfolded protein with the help of the chaperone, but in these conditions, in metabolic alteration, mutation, viral infection and chemical insults, the protein folding demand exceeds more than the capacity of protein folding of the endoplasmic reticulum, and this is known as endoplasmic reticulum stress. Whenever endoplasmic reticulum goes into the stress, this will result into the either adaptation, in which there is decreased protein synthesis, or increased production of the chaperone, and this will ultimately result into the mature folded proteins. Another mechanism is whenever there is endoplasmic reticulum is failed to adapt, then this will stimulate the mechanism of cell injury by apoptosis. Now we will see some of the common examples of disorders of misfolded proteins. This is the mnemonic we use for them: alpha 1 cat FC. Here we will see the disease and the affected proteins only. We will cover it very briefly. Alpha 1 antitrypsin deficiency, the affected protein is alpha 1 antitrypsin. In cystic fibrosis, affected protein is CFTR. Affected means misfolded protein. In Alzheimer's disease, A beta peptide. In Tay-Sachs disease, hexosaminidase beta subunit. In familial hypercholesterolemia, LDL receptor. Creutzfeldt-Jakob disease, prions. So all of these proteins disorder are the examples of misfolded protein disorder, in which endoplasmic reticulum goes into the stress and unable to meet the demand of a folding of misfolded protein and result into these diseases. Now one of the very important mechanism of cell injury we will discuss, that is oxidative stress or free radical injury. So what is free radical? Free radicals are, or we can say, they have one unpaired electron in their outer orbit. These unpaired electrons are highly reactive and they cause damage to the adjacent molecule like proteins, lipids and carbohydrates, and they result ultimately into the cell damage. Normally free radical generates during the cellular reactions. These are also known as reactive oxygen species, and there are multiple free radical scavenging mechanisms takes place in intracellular environment, and they keep a check on the level of free radical and maintain the low level of reactive oxygen species or free radical in intracellular environment. Failure of these scavenging mechanism result or increase overproduction of the reactive oxygen species will result into the oxidative stress. This can be seen in cell injury, cancer, aging, neurodegenerative disorders and various inflammatory process. Some of the examples of free radicals are hydroxyl free radical, superoxide, hydrogen peroxide or peroxynitrates. Now we will see briefly the mechanism responsible for the generation of free radical. In free radical generates in redox reaction, in radiation exposure, inflammation by NADPH oxidase, enzymatic reaction in which CCl4 convert into CCl3, transition metal in Fenton reaction in which iron involved, and generation of hydroxyl free radical. So these are the mechanisms responsible for the generation of free radical. Now we will see whenever there is an injurious stimuli, which could be radiation, toxin, chemical, whatever, result into the increased production of free radical. Free radical, okay. There are some scavenging mechanism as we have discussed, like superoxide dismutase enzyme convert these superoxide free radical into water. Hydrogen peroxide decomposed to water, while glutathione peroxidase and catalases. These mechanisms are responsible for the removal of free radical, or they maintain the low level of free radical in normal cell. What are the pathological effects of free radical? Lipid peroxidation is one of the most important pathological effects of free radical. Free radical attack the double bonds in unsaturated fatty acid of membrane lipid and result into the membrane damage. They also increases or result into the protein modifications by oxidation of amino acid or formation of covalent protein bond and oxidation of protein backbone. So protein modification caused by free radical by three mechanisms: oxidation of the amino acid, formation of covalent protein bond, and oxidation of protein backbone. This will result into the breakdown misfolding. Free radical also causes DNA damage. Single and double stranded strands break in DNA and ultimately result into the mutation. There are some substances known as antioxidant. They basically decrease the production of free radicals or increase the scavenging of free radicals. Some of the examples are vitamin A, C and E. So this is for this lecture. In the next lecture we will see about the clinico-pathological correlation or some of the examples of cell injury. Thank you.